Next speaker is Jonathan Kimmelman from Montreal in the Biomedical Ethics Unit. This session is really a continuation of Kathy's topics of trying to figure out how to design first-in-human trials in terms of preclinical data, natural history studies, and the ethics of trial design and the patient's perspective. So thank you, Jonathan, for agreeing to participate.
Well, thanks to the organizers for having me. It's wonderful to get a break from the frigid Montreal temperatures. Okay, let's get started here. So first, I just want to start with a disclosure. I serve as a DSMB member on an early phase clinical trial of ultragenics. My remuneration is less than $5,000 a year. So, okay, so I think it's helpful to start an ethics discussion by going back to some really fundamental basics, or at least the way that I view the ethics of clinical research in general, but in particular early phase research. So there's sort of two kind of canonical issues that we think a lot about when we do clinical research. The first one is something everyone here in the room already knows and already thinks about. When we do research in patients, we are using patients as reagents, as research reagents. That's different than in geology, where rocks are used as research reagents. Patients feel pain, they suffer, they have interests, and so accordingly, much of what we think about in research ethics is how to create a sphere that is protecting the human subject from undue incursion from the goals of science or the goals of the researcher. Okay, everyone kind of knows that. There's a second aspect of research ethics that I think not as many people spend as much time dwelling about, which is that the very reason why we do research on human beings is to generate information so that downstream users can use that information. And accordingly, we want to make sure that that information doesn't get adulterated in the process of generating that information and taking it up into healthcare systems. Now, there are three parts to this ethical component of human research. The first is that the individuals who do the research are highly trained and specialized individuals who there's a substantial public investment behind them. All of them have gone through medical schools, public education, et cetera, to develop their skills. And so if we put them on research, we want to make sure that those are productive lines of research and that there's no opportunity cost on that we're giving up by putting them into lines of research that aren't necessarily going to teach us something. Secondly, there's the issue about patients. The information we generate in clinical trials is ultimately going to be almost literally ingested by patients downstream in making life and death decisions in the clinic. Again, we want to make sure that we have the integrity of information, reliable information to make those kinds of life and death decisions. And the third issue is especially important in the context of gene transfer. We're talking about potentially very expensive products. Our healthcare systems or someone is going to have to pay for those. And we all know that U.S. healthcare system probably has the highest proportion of its GDP given towards healthcare expenditures. So if we are going to pay for these interventions, we need to be very confident that the information about safety and efficacy is highly reliable. So together, those are sort of two core reasons why we need to think carefully about the way we do research. Number one, protect the human subject. But number two, make sure that there is the appropriate screening and filtration systems on the generation of evidence so that we have reliable information to make life and death decisions. Okay, that's going to help frame what I'm going to talk about now in terms of some of the ethical issues. But first, I want to talk briefly about some of the concepts behind early phase research. So this is the way that I think about the goals of early phase research. All drugs, gene therapies, et cetera, are born guilty, and through the arduous process of clinical development, they're proven innocent. They're proven to be safe and effective. So the presumption is they're not safe and effective until they go through the rigorous vetting process. We can think of that process as having two basic steps. The first step is to identify the materials, practices, and beliefs that we need to combine with some substance, a vector, in order to elicit its therapeutic activities. So if we're talking about cell therapy for ALS, for example, those cells don't do anything unless we know exactly what dose to administer, what kind of immunosuppression regime to give, what kind of timing to administer in the progression of ALS, et cetera, with that intervention in order to elicit its activity. And so that's largely the task of what's going on in the so-called learn phase of clinical research, early phase research. And just to point out that a lot of those different aspects that we're trying to learn are variables that take on different values, and our goal is to sort of find as close as possible to an optimum value So we want kind of a near optimum dose before we advance into later phase clinical research. And ideally, I'm going to talk about later, we want to identify the lower bound on that dose to elicit activity. Okay, so we have identified the materials, substances, practices to combine with a substance. Our next task now is to take that intervention ensemble and put it into rigorous clinical evaluation, typically in randomized control trials. Okay, so that's going to help us to understand some of what I'm going to talk about now when I talk about these sort of three key ethical challenges in early phase research. First question I want to address is the question about how we decide when to initiate early phase clinical trials. Well, it's fairly simple in a sense. It's all about risk and benefit, where we understand benefit as the prospect of direct benefit to a volunteer, plus, if any, plus the benefit to society in the form of advanced knowledge. Now, in the realm of small molecules and even large molecules, FDA is not all that particular, not necessarily that careful about scrutinizing the quality of evidence suggesting benefit or social advance. They're quite explicit about that in their guidance documents. That's not the case in gene and cell therapy. There are a number of different guidance documents, Kathy alluded to them, that speak not only to the requirement to demonstrate potential efficacy in preclinical models, but that also talk about the quality of the design of those studies, the use of blinding, appropriate controls, et cetera. So there clearly is some kind of scrutiny that's going on about the prospect of benefit at the point of first-in-human trials from the standpoint of the FDA. Now, there have been cases out there, I'm at least aware of this one case, where there was a decision to launch a first-in-human trial, despite the fact that preclinical evidence that was available prior to the launch suggested that the particular product was not effective, in this case for spinal cord injury. So it's unclear exactly how careful the vetting is, but nevertheless, there is some kind of vetting. The larger point that I want to make is that decisions about the ratio of prospect of harm versus prospect of benefit are ultimately ethical judgments. FDA is very clear in delegating those kinds of ethical judgments to ethics committees, to IRBs. What's more, their agenda is not to advance the science, their agenda is to regulate drugs. And accordingly, judgments about the scientific value of a study are going to be judgments that IRBs are going to make. The reality is that many IRBs, it sounds like the one at CHOP is maybe a bit different, don't spend a lot of time really carefully vetting the preclinical evidence. It's very esoteric, it's very complicated material. And so the challenge that we have in highly intensive, very sort of science-intensive areas like gene transfer is that you have a process where IRBs are supposed to be vetting, overseeing, refereeing judgments about risk and benefit, and yet IRBs are often not really well equipped to do the careful analysis and scrutiny of the preclinical evidence to make those judgments about the prospect of direct benefit as well as social benefit. I've argued elsewhere about how we ought to fix this problem, but I do see this as a constant challenge in early phase research, including in gene transfer. Another recurrent challenge in the context of early phase research is the question of whether or not we consider exposure to new interventions to be therapeutic. I want to suggest that there are two reasons why we ought to tend to resist calling access to interventions in early phase studies therapeutic. The first is based on principle. I started off by saying that the very task of early phase studies is to identify the materials, practices, and beliefs that we need to combine with an intervention to elicit its activity. Well, at that point, we don't even know how to use the intervention, much less that the intervention is effective. And I think for those reasons, I think we ought to generally, although there are exceptions, be dubious of claims that there's a prospect of therapeutic benefit in first-in-human clinical trials. There are exceptions, and I can talk about this later. The second issue I want to point out is that there really isn't necessarily good evidence to suggest that participation in early phase trials is at a population level therapeutic. Of course, there are cases, there are anecdotes, there are exceptions. Kathy's described some of these. But nevertheless, we know that most interventions put into early phase research fail to vindicate the efficacy and safety through the process of clinical testing. In my own research looking at cancer, small molecules and large molecules, so not gene transfer, the fraction of patients who go into phase 1 studies who receive a drug, who have the right indication, who have the right sort of medical diagnosis, and who receive the right dose, a dose that will ultimately be approved by the FDA, is about 1 in 70. So that's a really relatively low odds of receiving a therapeutic intervention in early phase studies, given that the risk is somewhere on the order of 10, 15% grade 3 to 4 adverse events. That to me does not sound necessarily like a very clear case of a good, favorable risk-benefit balance in terms of a claim of therapy. So generally, and again, there are exceptions, I would say that we ought not to think about participation in early phase studies as therapeutic. Let me now move on to some recurring challenges in terms of how to design early phase studies. Again, I said this task is to identify the material practices and beliefs, to combine with the intervention to elicit its activity. One of the key tasks is to define the lower boundaries on those various elements in the intervention ensemble. What's the lowest effective dose? That's really, really crucial for healthcare systems to function properly. Here's just a kind of an illustration. Imagine we develop a stroke drug. This is not about dose, this is about timing. A cell therapy for stroke. We know that the cell therapy works when patients are about 4 hours after their hemorrhage or whatever their stroke event is. A patient shows up at the hospital 6 hours out, 8 hours out. Should we give them the cell therapy or not? Unless we have done the hard work of figuring out the window, the boundary on that window where the cell therapy is effective, we don't know whether it is cost effective and even beneficial for the patient to receive that intervention. So much of that becomes especially crucial where we're talking about expensive interventions to define that boundary. Much of the work of establishing the boundaries on those various elements is happening in early phase clinical research. And generally speaking, it's a lot easier to go from low therapeutic upwards, to escalate dose, to escalate timing, et cetera, than the reverse. So think about this. If you're a patient, and you know that an intervention is effective, would you participate in a randomized trial that was testing a half dose to see whether or not the half dose would be effective? Probably not. You would not go into that trial. You would be much more willing to go into a trial that is starting at a low dose and escalating upwards towards a therapeutic dose. So incredibly difficult to work backwards once you have an intervention that's out there into applications. Let me talk briefly about the challenge of informativeness. I've written elsewhere that trials, in order to be ethical, need to fulfill these 5 different requirements to inform healthcare systems. Here I want to just talk about one element in particular, the issue about reporting early phase research. There are any number of different challenges in terms of making sure that the information generated in early phase research is out there so that subsequent researchers and or healthcare systems can use it in patient decision making. This is a study that suggests that oftentimes in the context of at least cell therapy, there is somewhat of a delay between the completion of studies and the actual publications. There are any number of other sort of challenges that occur with reporting. And again, it's really critical that we recognize that if patients have participated in clinical research, we honor their sacrifice and contribution by making sure that we are promptly and completely reporting the results of their participation. Again, also in terms of making sure that we're efficiently using research resources. Okay, let me now close with just a couple quick remarks. I think everything that I said here is under a lot of challenge and strain increasingly in gene transfer, partly due to political circumstances, partly due to various trends in the ways that we are doing gene transfer research. And I want to close by just making 3 fairly quick remarks. So everyone here is familiar with the case of Mila, who received an oligonucleotide, antisense oligonucleotide for a very rare form of Batten disease. I think this case really illustrates some of the major challenges against some of the propositions that I advanced and about how we kind of oversee the ethics of early phase trials. So let's begin with the first question, when to begin clinical testing. Here's a case where the first inkling of doing a clinical trial from there to the first injection of the patient was about 1 year and 2 months. So an incredibly compressed timeline. The period from the first non-human animal toxicology tests to the first injection, 1.5 months. So a lot of pressure on, at least time pressure on the assessment of the preclinical evidence base for doing those studies. That's something that we're likely, as we move towards N of 1 studies, to continue to be challenged by. The second question is, do we consider Mila's access to this oligosense antinucleotide to be therapeutic or not? Is early phase trial participation therapeutic? there are a lot of ways in which, if you look at this protocol, it looks like research. It doesn't look like individual care. For example, there was IRB review. There was a rigorous informed consent process. This was FDA approved. There was all sorts of intensive baseline monitoring of the individual. On the other hand, there are a lot of other aspects that are not what we typically expect in the context of research. This did not go through a rigorous peer review process and selection. Unlike most research, there wasn't any kind of pre-registration of this in N of 1 clinical trial. There was no primary endpoint in the protocol. There was no clear data analysis plan. And there's not necessarily a clear path to license. So in many ways, this example really strains our traditional notions of what research ought to be. And third is the issue, I'm about to close. Third is the issue of informativeness. Which is that here we have a case study, and it's clearly a mostly spectacular outcome. It's great. It made all the newspapers. What we don't know are the number of other case studies that are out there in the literature. There's no mechanism here to pre-register case studies. So it makes it really difficult for us to look at this case and to draw reliable inferences about the utility of this particular approach. And I think, again, this creates major challenges. Here's my last slide. I just want to close with the idea that I think historically we've always had this idea that, okay, you can do early phase research, you can generate sort of, findings, maybe they're not conclusive findings, but we always can trust that there's going to be randomized controlled trials out there filtering those findings before we take an intervention and translate it into clinical practice. And I think for a number of different reasons, I think that is increasingly under strain. And I think that makes it especially crucial that when we do early phase research, including N of 1 studies, that we are holding them to the same standards that we would normally hold randomized trials to in terms of the transparency, in terms of the quality of reporting, et cetera. Done.
Great. Thank you.